this is Dr. Christopher Morosky, and this brief video is going to review the hormonal control of parturition. The content of this video comes pretty much exclusively from the journal article by Hamburg-Shields et al. With the same title, The Hormonal Control of Parturition, that was published in the July 2024 edition of Physiological Reviews, and you can use that for your reference. The learning objectives for this video are to describe key hormones involved in regulating parturition, including progesterone, estrogens, oxytocin, and prostaglandins, explain progesterone withdrawal and its role in triggering labor alongside increased estrogen activity, and to recognize the role of inflammation in placental peptides in initiating labor. So let's start off here by talking about the players. And the players can be kind of divided up into two groups, the uterotropins and the uterotonins. The uterotropins are hormones that either keep the uterus quiet and prevent the onset of labor, or hormones that will promote the onset of labor. We wouldn't clinically use these to induce somebody's labor, but these set up the uterus for eventual spontaneous natural labor. And then there's the uterotonins, and these are molecules that actually cause smooth muscle contraction and eventually result in changes in the uterus and labor and delivery of the baby. So the first hormone to talk about here is progesterone, also known as P4. Progesterone is made by the corpus luteum in the first trimester, and then by the placenta in the second and third trimesters. Progesterone binds the progesterone receptor with two isoforms, PRA and PRB. Progesterone is required for the maintenance of pregnancy, and progesterone really maintains the quiescence portion of pregnancy with minimal contractions. Progesterone is made in increasingly higher serum concentrations throughout human pregnancy, and importantly, placental production of progesterone is without any type of tropin stimulation or negative feedback like we see from the ovary to the hypothalamus and the pituitary. That doesn't exist with progesterone made by the placenta in pregnancy. So looking a little more closely at progesterone, you can see in the diagram on the left side that progesterone levels really do continuously climb from the first, second, and third trimester of pregnancy. There's no real fall off here in terms of serum progesterone. And then the production of progesterone in pregnancy, like I said, really isn't regulated by any factors. So you can see that on the maternal compartment, cholesterol will enter into the trophoblast or the placenta and be cleaved and converted to pregnenolone. And this goes all the way down to progesterone, but there is a CYP17 block here. There's no CYP17 activity, and so it ends with progesterone. That progesterone can move back into the maternal compartment and will do all the things that we're going to talk about progesterone doing in terms of pregnancy and labor and not having labor and labor. Estrogens are the next important hormones to talk about here, and estrogens are E1, which is estrone, E2, estradiol, and E3, which is estriol. Estrogens are made by the granulosa cell of the ovary, so that's really estradiol for the most part, and eventually by the maternal fetal placental unit where all of these estrogens are made. Estrogens will bind the estrogen receptor alpha, so ER alpha is the estrogen receptor, and that protein is made by the gene ESR1. Estrogen promotes uterine contractions and eventually primes the uterus for labor, so it gets the uterus ready for labor. It's a uterotropin. And estrogen is made in increasingly higher serum concentrations throughout human pregnancy, becoming the dominant hormone over progesterone in the late third trimester. Estrogen production is eventually tied into placental corticotropin-releasing hormone, which we're going to discuss later in this video. In these figures here, again on the left, you can see that serum estrogen levels do in fact rise throughout pregnancy, so they go up from the first, second, and third trimester, and you can see that they are mostly elevated, and there's a real upswing in how much estrogen is in the serum in the third trimester. Now, estrogen production requires the maternal fetal placental unit, so all three compartments. And so I want to just go through this quickly and in some detail here. So starting with the blue cholesterol in the maternal compartment, that will move into the trophoblast and become pregnenolone. That pregnenolone from the trophoblast can go into the fetal adrenal. And in the fetal adrenal, through a series of reactions, the pregnenolone is reduced down to DHEAS. That DHEAS by itself can move out of the fetal adrenal back into the trophoblast, where it is converted into E1 and E2, and that can move into the maternal compartment. And then back to the fetal compartment, that DHEAS can move into the fetal liver, where it's converted to 16-hydroxy-DHEAS. That molecule will move over into the trophoblast and be converted into E3, and that E3 will move into the maternal compartment. So you can see that the mom, the placenta, and the baby are all involved in the production of these 3 different estrogens during pregnancy. Now to talk about 2 of the uterotonins, the first one being oxytocin. Oxytocin is a 9-amino acid neuropeptide produced in the supraoptic and periventricular nuclei of the hypothalamus and secreted from the posterior pituitary. Additionally, oxytocin is also produced by the decidua and the myometrium of the uterus in pregnancy. Oxytocin binds the oxytocin receptor, which is a G sub Q protein-coupled receptor on myometrial cells, which result in myometrial contraction. And oxytocin receptors are upregulated by the estrogen receptor binding and are more concentrated at the fundus of the uterus than the lower uterine segment. Because when you think about delivery, you kind of want to shoot the baby out like a cannon. So you're going to put all of your gunpowder or squeeze more so up at the fundus, so that will push the baby out. There are various different prostaglandins, but the prostaglandins that we really focus on in pregnancy are prostaglandin F2 alpha, PGF2 alpha, and prostaglandin E2. So prostaglandins are synthesized from arachidonic acid by various, usually inflammatory cells. And in pregnancy, you want to think about activated macrophages and neutrophils in the placenta, the amnion, and the chorion decidua. Prostaglandin F2 alpha will bind the FP receptor, which is also a G sub Q protein-coupled receptor, causing myometrial contractions. And here, this is important, prostaglandin E2 will bind the E1 and E3 receptors, G sub Q PCRs, which also cause myometrial contraction. And it's important to remember that prostaglandin E2 and some of the other E's will bind the E2 and E4 receptors, which causes myometrial relaxation. So importantly, in terms of thinking about labor and uterine contraction, the uterine fundus has high concentrations of FP, E1, and E3 receptors at term. And importantly, prostaglandins also change the structure and organization of cervical collagen, causing the cervix to become more soft and stretchy, and clinically we refer to this as cervical ripening, or a ripe cervix. So the last thing to talk about here in terms of the players is inflammation. And there is now very good evidence that now supports that an increase in the inflammatory load of the pregnancy is one of the tipping points of moving from pregnancy quiescence to labor. This inflammatory load comes from various sources, including maternal stressors, which include the vaginal microbiome, and maternal corticotropin-releasing hormone, and some signaling from the fetus, such as fetal CRH, and last but not least, also intrauterine stressors. And increased inflammatory cytokines will activate local immune cells to synthesize prostaglandins from arachidonic acid, as I just mentioned earlier. Okay, so now that we know the players, let's start putting this all together, and we're going to start with the big picture. So if we sort of look at these 3 categories of the hormonal regulation of pregnancy and parturition, you can see that for most of pregnancy, there is this quiescent phase, which is really led by progesterone. And then eventually this moves over into labor, which has a latent and an active phase, and then after birth, there's an involution. The majority of pregnancy is in this quiescent state with low myometrial contractile activity, which is maintained by progesterone, or P4. This is the dominant hormone effector of ongoing gestation. The progression from quiescent pregnancy to latent or early labor is facilitated by a functional progesterone withdrawal and increased estrogen effect. The uterotonins, prostaglandins, and oxytocin are the prominent effectors of active labor and then eventually the postpartum state and the postpartum uterine involution. So let's start thinking about pregnancy in general, and we're going to talk about progesterone quiescence, which again is the majority of pregnancy. So progesterone binding its receptor PR, so P4-PR binding, inhibits expression of ESR1, which decreases the production of the estrogen receptor ER-alpha. P4-PR binding also inhibits the expression of the OXTR gene, which decreases production of oxytocin receptors, and progesterone binding its receptor inhibits the transcription response of uterine cells and tissues to pro-inflammatory stimuli, inhibiting prostaglandin synthesis. So for the pregnant state to progress from quiescence to eventually labor, there does need to be a progesterone withdrawal, but this is a little bit different in humans rather than other animals. So unlike other animals, and think of mice or sheep, humans do not experience a systemic withdrawal of progesterone leading to the onset of labor. Again, think back to that graph where you saw progesterone going up the entire pregnancy, even up to the moment of labor. So there's no systemic withdrawal. Instead, there is both a local withdrawal of progesterone through metabolism to less active metabolites, and also there is a functional withdrawal through modulation of progesterone receptor transcriptional activity, whereby in early pregnancy, progesterone binding its receptor occurs more with the PRB receptors, which have much greater transcriptional activity, and in later pregnancy, there is a greater PRA receptor binding, which leads to lesser transcriptional activity. So the combination of creating less active metabolites and modulating transcriptional activity to be lesser effectively creates a local and functional progesterone withdrawal, even though you don't see a systemic or serum concentration withdrawal in human pregnancy moving from quiescence to labor. So this really gets to this concept of a progesterone binding to a progesterone receptor block, or a P4-PR block. Also, this begins to incorporate this idea of the importance of inflammation to moving from quiescence to labor. The inflammatory threshold hypothesis for the control of parturition timing states that during pregnancy, inflammatory stimuli derived from multiple intrinsic, so think fetal development, placental senescence, and extrinsic, think maternal stress, intrauterine infection, factors contribute to a net inflammatory load impacting the uterine tissues. For most of pregnancy, the P4-PR anti-inflammatory block to labor prevents uterine tissue level inflammation. Above a certain threshold level, inflammatory stimuli induce P4 local and functional withdrawal, as I just talked about. Loss of the P4-PR block to labor allows a positive feedback pro-inflammatory state in the uterine tissues that induces active labor via increased contraction-associated protein gene expression and increased production of and sensitivity to the pro-labor uterotonins, prostaglandin and oxytocin. The timing of parturition is determined by the inflammatory load trajectory and the inflammatory load threshold for inflammation-induced P4-PR withdrawal. The threshold is normally reached at term, but can be reached earlier in pathologic states such as intrauterine infection. Another really important concept here has to do with estrogens, but to get there, we have to talk first about placental CRH, the production of cortisol, and then we'll get to estrogens. So as you can see here, we're looking again at the maternal compartment, the placenta, and then the fetal compartment. The maternal hypothalamus will make some amount of CRH, which is in the serum for the mom, and it will eventually get over to the baby. In the fetal compartment, there's also fetal CRH production, which has some activity. But importantly, as you get into the second and third trimester, the placenta starts to make its own placental CRH. Now, the placental CRH is one of those really important things that creates the first positive feedback loop leading eventually to the onset of labor and involves estrogens. So that placental CRH, the big purple CRH in the middle here, just below the trophoblastic cells, will move into the fetal compartment and in the fetal pituitary cause the release of ACTH, and that ACTH will stimulate cortisol production in the fetal adrenal gland. The cortisol will help mature fetal tissues, and so this process causes fetal tissues, which have started to grow and develop, eventually to mature and begin to do things. The cortisol then will also move back into the placenta, where this is the really important thing. In the placenta, cortisol has a positive stimulatory effect to increase placental CRH production. So this is the opposite of what we see with the pituitary and hypothalamus, where cortisol will negatively feedback on CRH and ACTH. In the placenta, the fetal-created cortisol will cause increased elevated production of placental CRH. This, therefore, is our first positive feedback loop of the onset of labor. That fetal cortisol will positively feedback to stimulate placental CRH production, and that placental CRH will again go back into the pituitary to make ACTH, more fetal adrenal cortisol, and then keep going in a positive feedback loop. That's the first positive feedback loop. Now, also overlaying this is the increased production of the estrogens that I was referring to earlier. So starting again in the trophoblast at CRH, the CRH, and you can see the positive feedback loop kind of going there, which is the same for the production of the cortisol. But that CRH will go to the fetal pituitary and cause increased production of ACTH. The ACTH in the fetal adrenal not only causes increased production of cortisol, but it also causes increased production of DHEAS, like we saw earlier in terms of the production of these estrogens. So that DHEAS will move back into the trophoblast at higher levels and then be converted into E1 and E2, which goes into the maternal compartment. And some of that DHEAS will go to the fetal liver and get converted to 16-hydroxy-DHEAS, again back into the trophoblast where it's made into E3. And that estriol will move into the maternal compartment. And so again, the positive feedback loop, while also increasing fetal cortisol to mature the fetal tissues, also increases the maternal, fetal, placental production of these three estrogens. And so we previously talked about the withdrawal of progesterone. Now let's talk about what happens when estrogen binds its receptor. So the P4PR withdrawal increases the ESR1 transcription of the estrogen receptor. The positive feedback loop of placental CRH results in greater production of estrogens as the fetal tissues reach maturity through increasing cortisol production. And so part of this is this idea of this fetal placental clock, where as the baby is reaching maturity and can survive born on the outside, the uterus is beginning to prepare for labor. Estrogen and estrogen receptor binding increase oxytocin receptor and prostaglandin receptor expression. Estrogen receptor and estrogen binding also stimulates proteolytic enzymes that result in cervical softening and membrane weakening, so think cervical ripening. And estrogen receptor binding increases production of gap junctions between cells to promote contractions. Okay, so you can see here what we have is moving from the quiescent stage of pregnancy, where progesterone is more dominant, you now get this withdrawal of progesterone and this undoing of the P4PR block, and moving from that quiescent stage into latent labor, now you start to see that estradiol and these estrogens, again made by the maternal fetal placental unit, begin to rise, and you start to see now an increase in myometrial contractile activity, and that's latent labor or early labor, which can go for days to weeks, and then we're going to talk about transitioning into active labor. So here really is where we can put it all together. And what we're looking at is the endocrine, paracrine on the left, and the neuroendocrine interactions on the right, which control parturition. And so beginning from the left on the top, multiple physiologic factors contribute signals impacting uterine tissues that are integrated into a putative inflammatory load. So think maternal stressors on the top there in the blue, the fetal placental development, and intrauterine stressors interact with and impact the total inflammatory load. In a threshold limited manner, inflammatory load induces progesterone receptor withdrawal, leading to the loss of the P4PR block to labor. And this includes increased local inflammation due to the loss of the P4PR anti-inflammatory activity, leading to increased production of inflammatory cytokines some of which, such as prostaglandin F2 alpha, are potent uterotonins, will cause contraction. The P4-PR withdrawal also increases expression of ESR1, which encodes the estrogen receptor, allowing circulating estrogens to increase response of uterine tissues to uterotonins. Again, prostaglandins such as F2 alpha and oxytocin. And all of this increases the extracellular matrix breakdown in the cervix and the fetal membrane, leading to cervical softening and membrane weakening, respectively. On the right, we see the final neuroendocrine positive feedback loop, which involves oxytocin amplifying contractions at the time of active labor. And so active labor is phasic myometrial contractions, cervical softening and dilation, and eventually results in membrane rupture. And this last part right here is the second positive feedback loop. It's also called the Ferguson reflex. And the Ferguson reflex is a neuroendocrine feedback loop where cervical and vaginal mechanoreceptors, their stimulation will increase posterior pituitary release of oxytocin. So as the fetal head descends in the pelvis, pushing on the cervix, causing the cervix to dilate and open, the pressure on the cervix and the vagina will feed back to the hypothalamus and posterior pituitary and release more and more oxytocin in a positive feedback loop that keeps active labor going. So that's it, everybody. That was kind of a quick review, but I think we did touch on these 3 learning objectives to describe the key hormones involved in regulating parturition, including progesterone, estrogens, oxytocin, and prostaglandins. We explained the progesterone withdrawal and its role in triggering labor alongside increased estrogen activity. And last but not least, we recognized the role of inflammation and placental peptides, such as CRH, in initiating labor. This last slide are our references, which include the Hormonal Control of Parturition article by Hamburg, Shields et al., and also the textbook for the course. I hope you found this video educational and helpful in preparing for class and for your studies. Good luck.